An enzyme is a protein that increases the rate of reaction by lowering the energy of activation. They have an active site unique to specific substrates for the substrate to fit in, fulfilling the lock and key hypothesis, where the lock is the enzyme and the key is the substrate. When the substrate enters the active site, it forms the enzyme-substrate complex, where the enzyme would change its shape slightly while the substrate binds itself to the enzyme. Then a chemical reaction would take place, breaking down the substrate into products, forming the enzyme products complex. The products will then leave the active site of the enzyme and the process would repeat itself. An example is catalase, which is the enzyme, and hydrogen peroxide, which is the substrate, that when hydrogen peroxide reaches the active site of catalase, would be broken down into water and oxygen. What is competitive enzyme inhibitor? In competitive enzyme inhibitor, they work by preventing the formation of enzyme-substrate complexes because they have a similar shape to the substrate molecule. When they fit into an active site, they will remain unreacted since they have a different structure to the substrate. Therefore, less substrate molecules can bind to the enzyme so that the reaction rate is decreased. Competitive inhibition are usually temporary and the inhibitor will usually leaves the enzyme. Therefore, the level of inhibition depends on the concentration of the substrate and inhibitor, since they are competing for places in the active site. An example for competitive inhibitor are cyanide, which acts as competitive inhibitor to the enzyme cytochrome C oxidase. This prevents the electron transport chain, the last part of cellular respiration, from working, meaning that the cell can no longer produce ATP for energy. Tissues such as the CNS and the heart that depends heavily on energy are particularly affected. For the non-competitive inhibitor, it interacts with the enzyme, but usually not at the active site but at the allosteric site. The non-competitive inhibitor reacts either remote from or very close to the active site. When the non-competitive blocker binds to the allosteric site, it changes the shape of the enzyme and also the active site, so that the substrate can no longer interact with the enzyme to give a reaction. Non-competitive inhibition is a type of enzyme inhibition where the inhibitor reduces the activity of the enzyme and binds equally well to the enzyme whether or not it has already bound the substrate. Non-competitive inhibitors are usually reversible, but are not influenced by concentrations of the substrate, as is the case for a reversible competitive inhibitor. An example of non-competitive inhibition could be cyanide, which combines dehydrogenase with the cytochrome enzymes responsible for the transfer of hydrogen atoms during cellular respiration. The non-competitive inhibition changes the conformation of an enzyme as well as its active site, which makes the substrate unable to bind to the enzyme effectively, so that the efficiency decreases.